Hi, everyone. We're just going to wait a few minutes while everyone joins. I'm excited to have a great session with you today. So we'll probably start in 5 minutes or less. I've got an eye on the attendee list, and I'll let you know when we're going to get going. Hey, everyone. We're just waiting a couple minutes to let people join before we get started. As with all Zoom webinars, you can feel free to participate via the chat, or you can submit questions using the Q&A box. And at the end of the presentation, we'll have some time for questions. But rest assured, if I don't get to your question during the time that we have here today, we'll go ahead and respond to you via email and make sure we get your questions answered. Also, throughout the presentation, we're going to be submitting a couple of polls in the chat, so feel free to respond. We'd love to see your answers. Okay, looks like we've got a good group of people here, so we'll go ahead and get started. Hi, everyone. My name is Mya Steadman, and I am a data scientist and a solutions architect here at Elucidates. And today I'm going to talk to you about our process to systematically improve data quality and model performance so that researchers can get the most out of their biomedical data. In this session, we're going to explore the specifics of Elucidates' NLP-based biocuration platform and how we apply the technology to generate model quality data sets for AI and ML use cases. So should be a great session. Okay, well, let's start at the beginning. In order to effectively train predictive models in drug discovery, you need large volumes of high quality, clean, and linked data. However, it'll be no surprise to anyone here that preparing these data can be a costly and time-consuming task. So as the drug discovery industry turns to AI and ML more frequently, there's a growing need for a process that can accurately and efficiently label these data at scale. So before we can dig into all of that, though, we first have to understand the factors that contribute to data quality and how we can improve them. Because, as I said, quality AI requires quality data. So at Elucidates, we're taking a data-centric approach, meaning that we're focusing our efforts on providing high-quality data that can properly train the model instead of spending the time trying to improve the model when you have inadequate data. And I want to acknowledge right from the beginning that there isn't a universal definition of what data quality looks like. So we're going to go ahead and try to define that as a starting point. So first, let's start with the intrinsic traits. And so intrinsic traits are inherent to the data, and they generally cannot be changed after that data has been generated. And so these intrinsic traits include things like experimental design, the measurements, and the metadata records. In an ideal world, all of the intrinsic traits would be high quality from the start, but we know that that's really the case and reality falls short. And so we have to focus on the extrinsic traits to improve the overall quality of our data set. And extrinsic data quality traits are those that are affected by the systems and the processes that interact with the data after it's been generated. And these traits can actually be greatly improved through curation. So let's pretend we're studying hepatocellular carcinoma. We've got 360 patients, which for our field is actually a pretty decent sample sized. Unlike internet companies such as Grammarly, which have access to billions of data points to train their models, we in the biomedical field don't have the luxury of enormous sample sizes. More typically, the data sets available to us are have sample counts in the hundreds. And so when the number of data points available is this small, any reduction in data quality can lead to a decrease in model accuracy. So we have two options to improve the accuracy of our model. We can either generate more data or we can clean the data that we have. And now it's not always possible to generate more data for a number of reasons. This could be due to the cost associated with additional data generation, both in terms of time and money, as well as the fact that generating new data could potentially introduce more heterogeneity into the study. And even when generating more data is an option, it's actually not the most effective one. And when you're thinking about how to improve the overall quality of your data corpus, the different parts of an experiment that you could potentially focus on, I understand that metadata may not be the first thing that comes to mind, but when we really think about it and we think about we look at the tools in our arsenal, it becomes clear that the biggest gains can actually be made by focusing on the metadata. So let's explore this a little further. Continuing with this example, let's go through the different parts where we could improve and try to improve our overall data set here. So starting with the experimental design, that's going to affect the desired outcome of the experiment. And I may not be able to answer my original question, but the resulting data can still be used for another purpose. So hypothetically, if we were to ask a different question of the same data set, I could make a small gain in quality there or in usefulness there. Moving on to measurement quality, poor measurement quality can't really be recovered from. Normalization will only get you so far. And if you don't trust the accuracy of a measurement, you're not going to waste your time using that data set. So again, assuming the best case scenario, we can only improve the data slightly by focusing on the measurement quality. So similarly, poor metadata records and poor metadata quality can severely reduce the usefulness of the data set, and it can ruin a model accuracy. However, as long as there are unstructured records, like the text of a publication or an ELN, the metadata can actually be improved at any time. And we already know this works. Improving the accuracy and the completeness of the metadata by cleaning the labels has been shown to improve model performance more efficiently than increasing sample size. And so if you look at this plot here, ideally you'd want to be in the top left corner with really nice clean labels and very few samples, right? That would be the best, but as I said, reality is often short of ideal. And so you kind of have two options, right? You can follow the orange line on the bottom here, and what you'll see is even with noisy labels in few of them, you have the lowest accuracy of our test, only around 67% or so. And even if we more than double the number of samples that we put into this model, as long as the labels are still noisy, we're barely gonna cross that 70% threshold in terms of test accuracy. However, following the blue line, when we feed 100 clean labels, so with accurate metadata, really nice and complete, even with fewer samples, our test accuracy is much higher right from the start. And that just continues to get better as we increase the number of samples that we provide, as long as the labels are clean. And so fueling models with clean and linked data can lead to significant improvement in performance in our AI systems, especially relevant for predicting long tail problems. So that means when you have limited data or less than 10,000 samples, and like we said, that's the case for the biomedical field is often very sample limited. Now, all that being said, data curation is incredibly time consuming, and the tools that we have to do so are lacking. In fact, as data scientists, we spend 4 times as many hours preparing the data to be used in ML initiatives than we actually do running the models and gaining insights. And while I don't think that's necessarily a surprising statistic, what I do find interesting is that almost all of the research and development efforts around AI are focused on creating resources and tools for training and running models, while very few advancements have been made to improve the data preparation process. And that's the part that takes far longer. That's where we at Elucidata are focusing our efforts, is we ask the question, what can we do to make public data more usable? And the approach that we took was to apply data-centric NLP models to fill in the missing annotations and standardize the metadata terms. And then we use data engineering to convert all of the varied file formats into one consistent file type. And we store it all in a structured data schema. So kind of going back to that original scenario, studying hepatocellular carcinoma, I want to elaborate on how we apply our technology to public data. And so to fuel that study, we went to Geo and we found this gene expression profiling data set. And we downloaded the data and the series matrix file that were containing the metadata. And I would assume that most of you here are probably aware of the state of affairs on geo. It's worth mentioning that we looked at this quite closely, that in any given data set on a repository like geo, about half of the annotations, the sample level annotations, are actually missing. And of the ones that are present, there's only about 2% harmonized, meaning that, from data set to data set, we're using consistent language. And while this may be annoying, but not prohibitive, to clean up one or a handful of studies, the amount of time that data scientists collectively spend on cleaning and wrangling data is enormous. And so this is where Poly comes in. We've largely automated the standardization and harmonization pieces using connectors. And connectors are, they act as scientifically validated pipelines that use our models to harmonize the metadata with little human intervention. And so we've taken the messy and inconsistent data from sources like geo. We've applied our curation process to fill in the missing annotations and our standardized ontologies. And we do this for over 20 different metadata fields at 3 different levels: the dataset level, or what we think of like the publication title. So that's the top level, the sample level, which is going to be the individual columns within a data set, and even the feature level. So that's the rows within a data set. And what you end up with is our clean metadata that's linked directly to the data matrix in a single consistent format, and it ends up significantly reducing the noise that's coming in from that data set. I want to take a minute now to go into a little bit more detail on how the curation process works. So first, we give an uncurated data set to our generative AI models, which does a first pass at curating and enriching the metadata. The next step is we'll have our expert curators come in, and they actually use a curation app that we've built to review the annotations that were assigned, the labels that were assigned by the model. And this is an active learning step. And during this active learning QC, the model is improving performance as the curators correct the previous mistakes. And the process has greatly improved the quality and efficiency of our curation process. So we still keep a human in the loop for QCing the annotations, but they're actually able at this stage to move a lot faster because they're being prompted by annotations that are provided by the model. And so once we've kind of gone through this cycle a few times and we're satisfied with the output of the labels, there's finally a built-in validation layer that assesses the quality of the data that's being uploaded by identifying errors and mismatches to preset data standards. So kind of automatically checking, making sure that the logic is sound. And then the data set is pushed to an omics atlas where it's indexed so that it is findable for easy use and reuse in reports and insight generation. Okay, so this is all well and good, and our process of curating and harmonizing data, it improves the quality of the labels, and the effects are more than just a more organized data drive, right? The high quality data is the bedrock of any successful ML project. And if you'll remember the ratio that I showed you earlier that said data scientists spend 80% of their time preparing data and only about 20% of their time gaining insights, I'd like to show you an example of what it looks like when this ratio is flipped. And so by starting with a solid data foundation, the typical target ID process in drug discovery is much more efficient. So we had the opportunity to work with an early stage therapeutics company that was studying AML. And rather than follow the typical process for target ID, which relies on running a large number of wet lab experiments to generate proprietary data, and then repeating those experiments to confirm their results, they wanted to leverage the vast amounts of AML data that were available in the public domain and supplement those data with targeted and really thoughtfully designed wet lab experiments. And so together, our computational biologists and data scientists and curators and their wet lab biologists set out to identify differentiation targets, differentiation-based targets with a data-centric approach. And the hypothesis was that patients with a higher proportion of cells in an earlier stage of differentiation would be more likely to respond effectively to treatments. In order to test that hypothesis, we first needed to gather all of the relevant data together and bring it onto Poly to be curated and harmonized. And now these high quality data that made up our AML omics atlas became the foundation for training the patient stratification model that was then used to predict differentiation targets. So I'm going to tell you this story now. So I mentioned that the first step was to assemble a corpus of multi-omics AML data that could give us a holistic picture of the disease. All in all, over 10,000 data sets from public sources, as well as in-house generated data, were aggregated. And we made the decision to use multi-omics data for a couple of reasons. The first being that the variety would make the model more robust and generalizable, because the goal of this project, again, was to find targets that would translate to the clinic, not just make a good story for a journal article. They needed to be very practical targets. And the second reason was actually quite practical. There just wasn't enough data from a single data type that had the cell type information that we were looking for to properly train a model. And so in order to use the data for a patient stratification model, we needed to know the cell types that were present in that data set at the sample level. And if the author hadn't provided that information, the data set couldn't be used. So from a very practical standpoint, we needed to look beyond just one data type to get the volume that we needed to be confident in our predictions. However, bringing together multiple data types presented a new challenge, which was how could we use all these different data types together? And instead of trying to normalize or come up with a complicated algorithmic way to integrate these data, we instead transformed the data from absolute expression values into a ranked order of the genes in each of the data sets, assigned some weighting to each data type, and then these ranked values per gene were actually used as the input to the model. So the next step, we're going to get really into it now, was to define the genetic signatures for each stage of cell differentiation. So the model was given the cell types and the ranked genes for each of those cell types from each data set. The model then made comparisons of gene pairs to classify each cell type. So in very simplistic terms, the model is rule-based. And the first rule says whenever the expression of gene A is more than the expression of gene B, then you assign this sample to class A. Another rule says that if the order changes and gene B becomes more highly expressed than gene A, then the sample then gets mapped to class B. So then we build on these rules. So instead of just simply depending on a single pair, we're doing this exercise on a huge number of pairs to get the ranking of genes for each cell. So I might be dating myself here, but I like to think of this model like one of those Cosmo quizzes, or like a BuzzFeed quiz where you're given a series of questions and then you find out what kind of cell you are, depending if you get mostly A's or B's or C's. But however, the pairwise comparison, it's not quite enough to define the cells. So instead of simply taking the decisions from each of these pairs, we also do some modeling on top of those decisions. And so what we get as an input is a binary construct, right? So if the rank of gene A is more than B, we call it 1, otherwise it's called 0. And we'll do that for all of the rules that we had, all of those binary comparisons. And so we get a matrix full of zeros and ones, and we do modeling on top of that in that binary matrix. Okay, so now that we have our defined genetic signatures for the cell types that are associated with each stage of differentiation, we took a large population of AML patient samples, and we asked the model to group the patients into cohorts based on their cell types. And what we found is that it split the patient population into two groups, one that consisted of a higher proportion of early stage cells, and one that had a proportion of a higher proportion of late stage cells. And what we saw, as hypothesized, was that the patients with the higher proportion of early stage cells, so cells in the earlier stages of differentiation, had better clinical responses than those with mostly late stage cells. This was obviously very exciting, but again, the goal is to get to targets that translate to the clinic, and we're not quite there yet. A little bit further to go. So then we took these two patient cohorts and we wanted to see what was the difference between these groups on a genetic level. So we actually performed differential expression on the patient population themselves. So we did differential expression analysis to get the list of differentially expressed genes for each of the cohorts. Next, we refined those genetic signatures using transcription factor enrichment analysis and network analysis. And then that was our refined genetic signature. Then we went one step further. And from these genetic signatures, we filtered down to a prioritized list of gene targets. And we did that based on both the drug ability score and some literature review, or what we actually built was a support vector classifier. And we we collaborated and used our context and the information that we had internally from our own expertise to decide whether or not a gene target would be practical to follow up on. And then we used this prioritized list of gene targets to create a panel. And we use that panel, if you're still following along with me, to screen cell lines for high expression of these gene targets, right? We need to identify the cell lines that create these genes that really segmented these two patient populations. And the theory being that the cells that highly expressed the genes present in these panel would be responders to the treatments. So our colleagues in the wet lab, they screened a very large panel of cell lines, and we measured the expression of the genes in our panel that we created. And we were able to identify 2 cell lines that we predicted would respond to inhibitors, and one that wouldn't respond as a negative control. And this is really exciting for us because now it was time to test out these predictions with inhibitors. And as you can see, in these plots over on the left, you're looking at cell viability plots. The DMSO is in blue, and our treatments, our tool compound, 2 different treatments, 1 control treatment, and 1 inhibitor is in gray. And as you can see, when we tested these cell lines with the tool compounds, the viability of the cell lines with the positive signatures, these graphs on the top, were significantly inhibited. While the cell lines with the negative control or the negative signature, the graph on the bottom here, the viability was not inhibited. And so this was obviously a very exciting result because we were able to go from a hypothesis to 2 identified AML targets that then responded to tool compounds. When we did this whole project relying mostly on public data rather than data that was generated de novo. And this was thrilling. And this result would have been exciting enough. And although just identifying these targets were a huge success, the timeline that it was done in, I think, makes it even more impressive. So the Gantt chart on the top is showing the typical timeline for a target ID project. And that typically lasts, depending on how efficient or what your starting place is, how many resources you put on it, around 12 and 24 months. Obviously that varies a lot, but just as an estimation, from hypothesis to a validated target, that's the 1 to 2 year process. And we were able to complete this process in just over 6 months, and with far fewer resources, both in terms of time, employees, and actual expense. Wet lab experiments are very expensive. And so to kind of wrap this up, I'm also happy to tell you guys today that our colleagues have gone on to file an IND for these targets, and I know I am, and all of the team here at Elucidates is eagerly awaiting that clinical data to really see what can come of this project. And with that, I just want to say thank you for your time and attention. I'd like to thank the team at Elucidata for helping me for doing this work and helping putting this presentation together, and our colleagues for allowing us the opportunity to share their story and work with us together. And with that, I'd like to, if there's any questions I could answer, I'd be happy to. Just please populate them in the Q&A. And thank you for your time. Oh, thank you for the applause. And if you don't feel confident or you don't want to ask a question right now, you can reach out to me or me directly. It's mya.steadman@elucidata.io. I'd be happy to answer any follow-up questions. You can book a meeting with me, reach out to me on LinkedIn. I feel like I should have a like and subscribe below. But again, thank you so much for your time. And I'd like to, oh, let's see, we did get one question. Right, that's a great question. And so the machine learning part takes longer because if I jump back to that Gantt chart, when we're looking at actually the non-poly part, when we're actually doing this, is you have to do the model's accuracy is going to be worse when the data you're feeding it isn't as clean. So you may have to do more iterations of training. You may have to kind of retool the model itself. You may need to go out and actually collect more data. However, because with poly, you're starting with that curated clean data, the labels are more accurate, and therefore the model is going to reach a higher accuracy and better predictions faster and with fewer samples. So that's why the model, the ML part is shorter when you're starting with a more solid foundation. Which expression unit was used to rank genes? Oh, so I am going to have to take that question offline. I do not remember off of the top of my head. I think it's going to vary based on the data type that was used. So the whole reason that we translated them to a ranked order of genes is so that we wouldn't have to rely on the absolute expression value. So I think it would vary based on the different data type that we were putting in. But I can definitely get back to you on that question. We'll take your email down and I'll make sure you get an answer to that. Looks like there's one more question, and I will stay on for a few more minutes here, but as people drop off, I just want to say thank you. Okay, for data harmonization, if some data fields are missing, how does Poly recover these missing? So not imputation. This is a great question. Thank you for asking this. We're not wizards. We're not imputing missing data. So what we're doing is when you look, when you're downloading a data set from Geo and you look at the series matrix file, those data may be missing from that table. However, the field value, the value that we're looking for, must be present in the unstructured text. So in the case of something like Geo, our data set from Geo, we're going to go and we're going to look at the abstract for that information. We're going to look in the text of the publication for that missing information. We're going to look in the supplementary files, right? And we're going to comb through and try to fill in those missing gaps, but it has to be provided from the author. So we're not imputing any data. Now, I'd like to just add on that in addition to going through the unstructured metadata records and filling in that data from those unstructured records into a structured tabular format, if the field is truly missing, right? So if data set A has a tissue present and data set B is not a tissue, doesn't have a tissue present, we're not going to leave it blank. We're going to intentionally go ahead in data set B and annotate that as none. So you can always be confident that if a field is missing, it's not because it was missed, it's because it's not present. So it'll be intentionally labeled as none. But yeah, to answer your question directly, we're not imputing values. It has to be present, it has to be provided by the authors. We use a variety based on the use case. This particular model for the use case that I walked through today was, is it on this page? Yes, it's actually a support vector classifying model. So we built that binary matrix based on the rules, and then we used that to do support vector classifying. But we're not limited to that model. That was the one that we chose to use for this particular use case. But if you work with us on what we call an insights generation project, we would obviously use a model that would be applicable to your use case. You know, we have expertise using all the different varieties based on the use case here. But okay, I am not seeing any more questions coming in. Again, I will just put my email in the chat here. And if anyone else would like to reach out to me, I'd be happy to answer any additional questions that may come up. And Yeah, again, thank you for your time, and again, thank you to the Elucidata team and our colleagues for the opportunity to present those stories. So I hope everyone has a great rest of your day.